complete. So good morning, everyone. Welcome to one more Athena talk organized by the Hellenic Mediterranean University in Crete, Greece. It's a great pleasure to have with us, Professor Danai Venieri. I will present a bit our today's presenter. So Danai Venieri is a professor in School of Chemical and Environmental Engineering. At the same time, she's a vice rector for finance and development in the Technical University of Crete, one of the most prestigious institutions that we have in Crete and in Greece in general. He has a bachelor degree from the University of Patras in the Department of Biology, then a PhD from the University of Patras from the Department of Medicine in 2005. Her research interests include applications of novel disinfection techniques. I think that her talk today is going to be around this. You can see also the title of her talk: dispersion of antibiotic-resistant bacteria, biodegradation of pollutants using various microorganisms, assessment of microbiological quality of the aquatic environment, and much more. Her work has received more than 4,400 citations, and her age index is 32. Today, Danai is going to speak to us about photocatalytic applications for disinfection purposes. We are very glad that we have Danai as a colleague, as a friend. And the floor is yours, Danai, for the next 40 minutes with our participants. And also, I would like to thank the participants for their participation today. Thank you, Danai, very much.
Thank you very much. Good morning, all. Kostas, thank you very much for the invitation. It's an honor to participate in Athena Talks. I've been collaborating with your university many years, so it is a pleasure to present some highlights today regarding the photocatalytic applications, as you already mentioned, for disinfection purposes. I know that the background of all of you might be quite diverse. Actually, I'm a biologist, as Kostas mentioned, surrounded by engineers. So today, I would like to highlight and give you some insights about the pathogenic microorganisms and recent trends regarding their inactivation during disinfection processes. You may see here the overview of today's talk. We're going to highlight some hints, some tips regarding disinfection, and we are going to focus on advanced oxidation processes as disinfection techniques. Furthermore, I'm going to present some results regarding the application and the effectiveness of photocatalysis and solar photocatalysis for the inactivation of variant microorganisms. We're not going to focus only on bacteria, as they mentioned in legislation, but we're going to see what happens, what's going on regarding tolerant microorganisms like viruses as well. And finally, I'm going to present some recent results regarding the application of such techniques in pilot scale applications for disinfection of water and wastewater as well. So to begin with, waterborne diseases, as we all know, even after the so long COVID period, documented worldwide, and their rapid transmission through the consumption of contaminated water illustrate the importance of effective inactivation of pathogens, including bacteria, viruses, and many others like parasites, protozoa, phage. I'm sure you all know that we are facing nowadays many diseases caused by contaminated water like cholera, cryptosporidiosis, diarrhea, malaria, typhoid. Recently, we had here in Hania some incidents of gastroenteritis caused by noroviruses. So we can all conclude that public health protection is urgent, and we always seek for new techniques for the inactivation of pathogenic microorganisms. So disinfection, yes, absolutely, is an essential and final barrier against human exposure to disease-causing microorganisms. You can see here at this table microorganisms detected in wastewater, not only bacteria like total coliforms, fecal coliforms, enterococci, salmonella, but also parasites are present like Giardia and Cryptosporidium. Both of them are mentioned here because they are also mentioned in current European legislation about the suitability of water, consumption of water, potable water. But we should never forget the presence and the virulence of viruses, like enteric viruses. These are present in environmental samples, seawater, potable water, and absolutely wastewater. Also, before beginning with any results or any experimental setups regarding photocatalytic applications, I would like to discriminate disinfection from sterilization. Disinfection involves a physical or chemical process that inactivates pathogenic only microorganisms, but without necessarily reaching the point of killing them. So the correct term to use when we are referred to disinfection is inactivation, not complete killing. On the other hand, sterilization is a rather extreme process under which the complete destruction of all organisms is achieved. So what happens regarding water and wastewater is disinfection, not sterilization. We live among different kinds, different groups of microorganisms. We live together. So the primary target of disinfection is the disease-causing microorganisms, not all of microorganisms present in the aquatic environment. And of course, we need to control their concentration to tolerable and safe limits for public health protection. As you may know, the common disinfection techniques include basically chemical compounds, filtration, or radiation. Each one of them has different means of action, like growth prevention of cell wall, destruction of cell wall, or even destruction of the genetic material. But we face many considerable disadvantages. For example, chlorination is well known for its toxic byproducts. Ozonation, the second best, is a very effective technique, but with high cost. UVC radiation is another option, but with many action limitations, specifically when we need to disinfect complex samples, like the one which is derived from a wastewater treatment plant. Also, when we deal with disinfection, there are many issues under consideration, like, for example, the type of microorganism tested in each case, or the initial concentration of microorganisms in our samples. Many conditions, pH, temperature, the potential of certain microorganisms to reactivate after disinfection. This occurs specifically when UVC radiation is applied. And last but not least, we should absolutely pay attention on the water matrix. For example, it is very much different when we need to disinfect water than wastewater. Turbidity, dissolved organic matter, as you may see here, make our life quite difficult. Generally, wastewater is much more difficult to be disinfected than water samples. And I will present some examples and some results regarding wastewater disinfection. Also, another issue, the type of microorganism. Most research studies deal with bacteria like E. coli and Enterococci. Both of them are mentioned in European legislation. But of course, in wastewater or in water, other bacteria may be included, like Bacillus, Staphylococcus. And those bacteria, of course, are not the same. They have different structures. For example, E. coli and Enterococci, which are mentioned in legislation, E. coli is gram-negative, Enterococci is a gram-positive. Enterococci is a broad group of microbacteria that includes species belonging to the gram-positive types of bacteria. I'm not going to get into many details, but because of their special structure, you can imagine, you can think that they behave differently in treatment processes, in disinfection processes, or they could be, some of them either sensitive or tolerant under different environmental conditions. Also, beyond and apart of bacteria, there are many other microorganisms included in aquatic samples. Phage, these are viruses which infect bacteria. But apart from them, there are other viruses which infect human, like adenoviruses or enteroviruses. Both of them are mentioned in European legislation, but they are not controlled, they are not analyzed regularly. So, and also the protocols regarding their analysis is quite difficult. And moving from viruses, there are also other organisms like parasites, Giardia and Cryptosporidium, specifically when the water is derived from surface sources like a lake or a river. So you can see the broad variety, the great variety of microorganisms that may be included in water and of course in wastewater. And just a few details about the kinetics of disinfection and the overall inactivation of microorganisms during a process. So inactivation is actually a gradual process that involves a series of physicochemical and, of course, biochemical steps. We usually use this Chick-Watson model that you can see here. And in the diagram, you can see the most common appearance of inactivation of certain microorganisms through time. So you can see the shoulder, the ideal first order reduction of the microorganisms, or the rapid inactivation. But I would just like to highlight the tailing off, the black line here, which means what? That many microorganisms, specifically the resistant ones, may be present even after a very strong, let's say, effective disinfection technique. This is the tailing off curve that you can see here, which means that there might be some remaining microorganisms. We think the most resistant ones after the process. So it depends. It depends on the type of the microorganism that we try to inactivate each time. if this if the case is that it is a very virulent one, it will cause a disease. Why? Because the virulent ones have a very low infectious dose. So you can think, you can see that it's a very complex matter, complex issue when we need to disinfect a water from a distribution system or wastewater for reuse applications. So at this point, and trying to explore new techniques, advanced oxidation processes seem to be a very good, let's say, choice, at least up until now. They have been recognized as an emerging group of techniques with high oxidation potential and biocidal effect on various microorganisms in aqueous samples. You can see here some examples like catalysis, ozone, or electrolysis, wet oxidation, phantom, photophantom. So what's going on with those techniques? The advanced oxidation processes, the ALPs as we call them, rely primarily on the in situ generation of highly reactive intermediate chemical species. A very good example is the hydroxyl radicals, which can cause eventually oxidative stress to the microorganisms. This term is quite important. I mean, when we hear the stress called to a cell, to an organism, this means that the cell will be damaged. And when the cell is damaged, then we can conclude that the microorganism is inactivated, including the pathogenic strains, of course. We are concentrated on the pathogenic strains. So this is the whole process generally is very simple and effective. It is actually at least certain aspects of ALPS are quite cost effective as well, and they can be applied for many different environmental applications. And actually, we think we can conclude that those techniques are quite sustainable. First of all, advanced oxidation processes, I would like to mention that they were primarily developed for the degradation of organic chemicals, organic pollutants. However, we have seen that during this process, they are also quite effective for the inactivation of different and virulent microorganisms. So today, I'm going to focus on a specific ALP, advanced oxidation process, the semiconductor photocatalysis, as you can see here, using titanium dioxide. This is quite a promising technique for microbial inactivation in many different matrices. So titanium dioxide is one of the most widely recognized semiconductor photocatalysts, triggering off the oxidative destruction and mineralization of a wide range of organic substrates and, of course, microorganisms. It is highly efficient with very low toxicity. This toxicity even in many times is quite absent, I should say. I mean, according to my experience in the experimental setups that have been applied here in my lab. It has also physicochemical stability and, of course, low cost. Now, what's going on with titanium dioxide? It is mainly active under the UV spectral range with a high required gap energy, as you may see here, 3.2, approximately 3.2 EVs, for excitation of the semiconductor. So under illumination and upon illumination, what happens is the generation of reactive oxygen species, the ROS, the so-called ROS, which finally, ultimately cause the oxidative stress to many different microbial populations. So in this way, we can achieve the inactivation of different microorganisms, even viruses, even phage. I'm going to show you some examples and some results later during this talk. So up until now, this process seems to be quite effective for the disinfection of even complex matrices. What makes this process even more attractive is the option to use those catalysts under visible light energy. During this process, researchers have found so far that if we modify the catalyst, the titanium dioxide, for example, modifying its structure upon metal doping, we can make this catalyst be active under natural solar light. So we have achieved to generate metal-doped titanium dioxide materials as catalysts, as photocatalytic semiconductors, with the hope, with the potential of them of being active under solar light spectrum. And of course, doping this catalyst by several metals broadens the region spectrum of these semiconductors towards visible light region, which seems to be quite a hopeful, let's say, technique for the effective and cost-effective, of course, inactivation of all different kinds of microorganisms. So yes, solar photocatalysis, I mean, with the use of natural solar light, is actually a very effective solution for disinfection of water and wastewater. And light source and a semiconductor material, yes, they do comprise a powerful duo, as we may say, that may offer several photocatalytic applications for, of course, environmental remediation. Now, what's going on during solar photocatalysis? I mean, what's going on or in the cells, on or in the microorganisms? I'm just highlighting this because viruses are not considered microorganisms. They do not possess the cellular structure. So they are considered quite different from all the others, bacteria, parasites, fungi, and so on. So you can see here in this figure the general mechanisms of action of sunlight for water and wastewater disinfection. And if you look closely, you can see that what's going on upon solar light illumination is the generation of those ROS, the reactive oxygen species, which ultimately cause damages, destruction to the enzymes, to chromophores, or even to the genetic material. So even if we don't use a catalyst, even if we don't use anything beyond solar light itself, specifically because of the presence of the UVA, can cause the generation of those ROS. But the whole process is quite prolonged. long treatment times are needed if we just use the natural solar light. And by prolonged times, we mean 1-2 hours. This is not considered quite effective. However, solar light is, let's say, the base in order to explore this process for effective and quick disinfection and activation of our microorganisms. So let's see what we can achieve with doped titanium materials. This first example deals with bacterial inactivation. Actually, you can see here that we deal with Staphylococcus aureus, not the well-known E. coli. If you search the literature, you will find numerous studies with E. coli disinfection, E. coli inactivation. Yes, this is the simplest bacterium, the very well-studied bacterium, the bacterium that is mentioned in legislation, but not only E. coli is present in water or wastewater. Many other microbial groups, types are present. So we always need to explore better conditions for the inactivation of other bacteria, even more resistant than E. coli. So Staphylococcus aureus, you may know this species. It can cause skin infections. It might be present in the water of a swimming pool. So we tried to explore those materials for its inactivation under simulated and natural solar irradiation. Of course, when we work with catalysts, and this is what my colleagues do effectively, of course, we try to explore the features and the whole characterization of the catalysts. Here you can see the XRD patterns and UVV's absorbance of those catalysts. You can see also here different dopants of iron, aluminum, chromium. So anatase phase is present in all samples. Also, we didn't observe any obvious diffraction peaks attributed to the oxides. And what is interesting, of course, is the UVV's absorbance. You may see that compared to the commercial P25, P25 is the pure titanium dioxide. So compared to that, there's a shift of the absorption edge to the visible light region. So according to those diagrams and to the overall characterization of the catalysts, there was a potential of using them under solar light, either simulated or natural. And actually, this was what we achieved. You can see here the Staphylococcus inactivation in using all those catalysts and the commercially available titanium dioxide, the P25. Of course, we tested many different concentrations of the catalyst. And just to point out here that the highest concentration of the catalyst does not mean that it is the best one. No, because the highest concentration of the catalyst, we're talking about slurry phase, leads to high levels of turbidity. So the transmittance of the light is prohibited. Anyway, you can see here that under the specific operating conditions and with the use of that concentration of catalyst under simulated solar light, we achieved quite successful, let's say, reduction of our bacterial concentration. We achieved 0 levels, as you can see in the diagram A on the left, but in many other cases, we saw the tailing off curve, as I showed you before, regarding the kinetics of disinfection. This verifies the fact that this Staphylococcus aureus, this specific bacterium, is quite resistant. So there is the possibility of remaining cells, remaining resistant bacteria after the treatment. However, we can see an almost 7 or 6 orders of magnitude reduction of our bacterium. Also here another detail, as you may see, we worked with really high initial bacterial concentrations. Those high initial bacterial concentrations are not normally found or detected in environmental samples. However, when we test, when we try to see if a disinfection technique or certain nanomaterials are effective, we always try to test high initial concentrations in order to see the level of effectiveness of each technique or of each nanomaterial that is used. And this is what happens under natural solar light, but again with the very high initial concentration. So at this point, you may see and you may recognize the fact that, yes, we do not have complete inactivation. No, this is, you can see here a very distinct tailing off period. There is substantial survival of resistant bacteria, resistant subpopulation. And of course, this comes as a result of protection by interfering substances, suspended matter in water, clumping, or genetically conferred resistance. Anyway, based on this diagram, we can see that there is certain inactivation under natural solar light, but of course with a specific catalyst and with fewer orders of magnitude. But of course, we should pay attention. This is a whole other issue when we work in the lab under conditions, under simulated solar light, and a whole other issue under natural sunlight. And this is what happens to the cells, to the bacteria, to Staphylococcus in our case. Here you can see some images. A and B present the intact cells, the Staphylococcus cells, and moving from C, D to F, you can see the destruction occurred, or the destructions occurred because of the presence and the interaction with the catalyst. So there is absolutely effects on morphology, cellular shape. The smooth cellular surface areas become ultimately quite rough, which means all those features, they mean that the bacterium is definitely inactivated. And here I would like to mention some details regarding the overall process of inactivation of microorganisms during photocatalytic techniques. You can see here the intact bacterial cells, and the whole process, which is a gradual process, begins with the interaction between the catalyst, nanoparticles, and the bacterial surface. Here, please pay attention, please observe that we are talking about nanoparticles. These are the catalyst, and bacteria. Bacteria are quite larger than the nanoparticles. They are in the scale of micrometers, many micrometers, I should say. So there has to be a certain kind of interaction, attachment, probably. And this attachment, this interaction leads to the morphological changes and deformation of bacteria. there the oxidative stress begins actually towards the outer cell wall components, and the whole biocidal effect is expanded towards the cytoplasmic membrane. So what happens is the increased cell permeability and outlet of intracellular components. If we have any leakage of the intracellular components, like for example potassium ions, then cell death, cell inactivation is achieved. And of course, if the overall process is prolonged, the destruction may be present even in the genetic material. The genetic material, which is not in nucleus, we're talking about bacteria in our case here, but this might happen. So the destruction of DNA, simply. And here you can see some images. This is, of course, this whole progress, this progressive inactivation is illustrated in those figures. This is a scanning electron microscopy regarding E. coli and Klebsiella pneumonia. E. coli is the well-known bacterial indicator. Klebsiella, just to mention here, is a very well-known, of course, bacterium, which demonstrates high resistance to antimicrobial compounds. And it is usually the cause of many hospital infections, at least nowadays. So if you take a look at those figures, you can see definitely the destruction of the cells in the case of E. coli and Klebsiella. But also, if you look closely, in the case of Klebsiella, in those red cycles, for example, in the first figure with the red cycle, you can see a pure hole in the cellular wall of Klebsiella, which means destruction of the outer layers of the bacterium, which of course means complete inactivation. And in the next figure, you can see the complete destruction of Klebsiella because of the interaction with the catalysts. And this is a very promising result regarding the inactivation of even the antibiotic-resistant bacteria, like that of Klebsiella. So just to give you a hint, to give you some details about the resistance level of bacteria, as I mentioned before, there's a great variety of them, the gram-positive, gram-negative, with different structures. What's going on actually is, regarding the resistant ones, or I should say the gram-positive ones, which seem to be more resistant, is their potential to survive even in extremely hostile conditions. Why? Because they are able to generate endospores, spores, like the spores of plants. So they can remain after treatment, or they can remain intact during treatment. So this is an issue with bacteria. So this is not, we don't have only E. coli. We do have many, many other bacteria with other features, structures, and potentials. What about protozoa? We don't hear them very often. However, all of them, specifically Curtosporidium and Giardia, which are mentioned in legislation, are present in surface waters. And they circulate in the environment in the form of cysts, oocysts and cysts, respectively, which are resistant forms, let's say, of their organism. Actually, protozoa are very, very persistent during disinfection. It's very difficult for us to set the conditions in a way in order to inactivate them. And actually, the best way to get rid of parasites, protozoa, this is the same thing. So the best way to get rid of them is to apply filtration. They are extremely larger microorganisms than all the rest of them. So the most effective technique approach is to filtrate water, or wastewater, if they are present. And of course, we should never forget viruses. We all remember the COVID period, COVID viruses. I'm sure we all know many details by now regarding viruses. The thing about them is that they are quite tolerant under stressed conditions. They do not have any cellular structure. The only structure available for attack is the protein of the capsid they possess. And at this point, we basically talk about enteroviruses and adenoviruses. Both of them belong to the group of enteric viruses. They're, of course, very virulent, pathogenic, absolutely. Complete absence is necessary for the safe and the suitable consumption of water. And what happens with viruses is that they are adsorbed onto the catalyst's nanoparticles. So there is this adsorption step. And because of this adsorption, there is the interaction and the final destruction of their structure. However, their inactivation may occur because of the other ROS, like the hydroxyl radicals, in the bulk phase of our solution, of our sample. So speaking about viruses, let's see what's going on when we wish to disinfect real wastewater samples and to test whether certain viruses, like the one that you can see here, bacteriophage MS2, whether this virus may be inactivated under simulated solar irradiation or natural solar irradiation. We used, and this bacteriophage may be used because it has the same, let's say, behavior with all the other enteric viruses. And this is actually an indicator of fecal pollution regarding the aquatic environment. So at this point here, you can see that, again, metal-doped titanium dioxide catalysts seem to be quite effective for its inactivation in wastewater samples. Those diagrams were generated upon application of simulated solar light, and I highlight the specific matrix wastewater because it is a very difficult sample. It is a complex sample. So special conditions are required for the disinfection of wastewater. It is not a mild procedure, like, let's say, disinfection of potable water. Here we deal with organics, nutrients, inorganics, interfering substances, which actually inhibit the overall process of disinfection. However, at least under simulated solar light, we can see here that those viruses were quite reduced during treatment. However, I should point out that prolonged treatment periods were required. I mean, 60 minutes for disinfection is quite a long period. But anyway, even in the images on the right, the TEM images, transmission electron microscopy images, we can see the destruction of the capsid. If we look closely, we can see the destruction of the capsid of the virus. But even the natural sunlight, under the natural sunlight, we can see a satisfactory, let's say, reduction of our population. I mean, almost 4 orders of magnitude, is quite successful. And actually, we achieved that result with binary doped catalysts, 2 dopants, manganese and cobalt, again, until during 60 minutes of treatment. Of course, if you see on the right, the rate constants, a linear fit of phage and activation kinetics, you can see that the rate constants are quite different comparing simulated solar irradiation and natural sunlight. Yes, absolutely. Under natural conditions, the overall process is quite long. Longer periods are required. However, yes, it is a promising result. We can achieve up until let's say 4 order of magnitude reduction of our virus. And speaking of virus and resistance and tolerance, what happens with antibiotic-resistant bacteria? This is a whole other issue. Nowadays, we do face the problem of antibiotic resistance in microbial populations in the environment because of the excessive use of antibiotics. Antibiotic is any class of organic molecule that inhibits or kills microbes, but an antibiotic is a metabolic product of another microorganism. Otherwise, we should use other terms like drugs or chemostatic drugs. Antibiotic, this is quite a strict, let's say, is a metabolic product of a microorganism against other bacteria, fungi, sorry, parasites, and so on. So there is an excessive use, broadness of applications, and an overall increase in antibiotics consumption, which leads to what? To the generation of resistance in microbial populations. Microorganisms have one goal in their life: to survive, to exist, to proliferate, and to survive no matter what. So they try to adapt in the presence of hostile elements like the antibiotics. So what happens is the generation of mutations in their genetic material. There is an increased acquisition of antibiotic resistance genes, another emerging pollutant, as we call it nowadays. Antibiotic resistance genes, which can, of course, go inside and travel inside microbial populations and generate the tolerance against different classes of antibiotics. So, a challenge is to see whether those techniques could be effective against antibiotic-resistant bacteria, like the Klebsiella pneumonia I mentioned before. So, what happens with this bacterium, which is highly antibiotic resistant and responsible for many hospital infections? And moreover, what happens with its antibiotic resistance profile after treatment in the remaining bacteria after treatment? First of all, those techniques seem to be quite effective. Those catalysts seem to be quite effective for the inactivation of Klebsiella, but just to highlight, take a look at the really higher concentration of the catalyst. In the previous slides I showed you, we used 25 or 50 milligram per liter. In this case, we used higher concentration, which means what? That antibiotic-resistant bacteria might be quite resistant against disinfection technique. And I underline might be because we cannot say one rule. We cannot have one rule for every single bacterium because of the great variety. Nevertheless, in this case, we saw that solar photocatalysis was quite effective, at least with the use of certain catalysts. We achieved an almost reduction of our bacterial of 7 orders of magnitude. But again, take a look at the tailing off of the curve. We're talking about an extremely resistant bacterial species. So there is this tailing off curve. There are remaining cells after disinfection. But of course, yes, the binary doped catalysts, manganese and cobalt, seem to be quite effective, at least compared to the commercial P25. P25, I'm just reminding you that it is the pure titanium dioxide without any dopant. And this is the interesting thing, just to show you how difficult and how complex is the disinfection process. You can see here the resistance profile of the intact cells of pneumonia and of Klebsiella and the resistance profile after treatment. The MIC that you see here is the minimum inhibitory concentration of each one of those antibiotics, which is required, this concentration, for the activation of, let's say, 60% of bacterial population. So, for example, regarding tetracycline, in intact cells, 32 microgram per ml of tetracycline were required to inactivate that bacterium. And after treatment, only 2, which means that that antibiotic-resistant bacterial species altered its profile after treatment. Since the bacterium required only 2 microgram per ml, means that it became more sensitive. So this is another benefit, but of course this is not the main result that we could get from every single bacterium or for every single antibiotic. For example, if you take a look at the ampicillin, we can see that our bacterium, Klebsiella, became more resistant or more sensitive or remained the same. So this is quite a diverse picture regarding the antibiotic resistance profiles. It's quite difficult to estimate what will happen in the remaining cells if the bacterium survives, the antibiotic-resistant bacterium survives after treatment. So this is, of course, a whole area of research. Many researchers deal with antibiotic-resistant bacteria. It is not quite easy. And of course, they deal with antibiotic resistance genes, which will be shown to you later. But, okay, those processes are absolutely, let's say, appealing. But how could we apply them in pilot scale applications? Solar photocatalysis is a very good choice. It could be applied in pilot scale applications, specifically in compound parabolic collectors. You can see here some images. This is the image of the collector. Actually, there are many of them in Plataforma Solar de Almería in Spain. And what happens is that those tubes could be supported by photocatalysts. I mean, we could support the photocatalyst into the glass tubes, and those pilot scale collectors could be applied for the inactivation of microorganisms under natural solar irradiation. And actually, those large scale applications could be applied, could be possibly replace biological treatment in wastewater treatment plants, especially in small, in limited wastewater treatment plants, offering a combined secondary and tertiary treatment. And actually, this is the case. This is our case. What we tried to explore in a pilot scale application, a compound parabolic collector in the campus of our university, Technical University of Crete. And specifically in this case, we tried to see whether viruses, enteric viruses or resistance genes could be eliminated during treatment, after treatment. In the case, of course, of real wastewater samples. And this is the collector. You can see images, photos of the collector in our campus. Of course, yes, I should say that we need sunlight and possibly good sunlight and high temperature. I mean, all those experiments were performed in temperatures from, I don't know, 27 to 30 degrees. But, and of course, here you can see, we should never forget the characterization of the catalyst. The binary doped catalysts in this case, different catalysts with iron, indium, aluminum, manganese, and so on. And of course, the UVV spectra. Here are the semi-images. Upon doping, we did not detect any specific changes in the morphology. And here you can see even more characteristics regarding the average particle size. I should mention here that the average particle size, yes, plays important role up to a certain extent regarding the effectiveness and the inactivation of microorganisms. And of course, the band gap energy here. Anything less than the 3.2 I showed you before, which is the band gap energy of titanium dioxide, seems to be a good choice. But, okay, yes, we did achieve those results, that characterization that you may see. I'm not going to delay you with this. Just take a look at the inactivation of E. coli and Enterococci. Those results are from lab scale experiments under simulated solar light. So you can see we could achieve complete inactivation even in 40 or in less than 40 minutes. Again, just to point out the high initial concentration and also the low concentration of the catalyst, 20 milligram per liter. Here you can see the rate constants. A more detailed, let's say, observation could reveal some differences between the performance, the behavior of E. coli than the Enterococci. In certain cases, they behave quite the same way, let's say. I'm just highlighting this because both of them are in the legislation, but Enterococci are gram-positive, which means that they might be more resistant in extreme environmental conditions. So this was obvious in some experiments. I mean, the higher the K, the value of the K, the rate constant, the more rapid is the inactivation. So in the case of certain catalysts, both bacteria behave the same way. On the other hand, for example, in the case of aluminum manganese, we can see that E. coli was readily inactivated more rapidly compared to the enterococci. However, the use of the pure titanium dioxide led to really prolonged periods for the overall inactivation. But what is interesting is to see what happened under natural solar light. Yes, we did achieve the reduction of our bacterial concentration, bacterial population, but in prolonged periods of time. But of course, it's dealing with natural conditions, experimenting under natural sunlight is far, far more different than doing the same procedure in the lab. So that's why you see the time 30 W that in the axis of the diagrams. This is the normalized, as we call, illumination time. It refers to a normalized solar UV power on a perfectly sunny day. It is quite different to work in the lab with the solar light right upon your sample, and quite different to work outside with natural sunlight. So in order to compare the results, there is the need to normalize somehow the time. Okay, details are not necessary here, but just the diagrams, both diagrams here are comparable to the previous ones. So it is quite promising to use such applications, such reactors for natural, the exploitation of natural solar light for the inactivation of such bacterial indicators. Again, you can see here the rate constants, differentiation between both bacterial groups. But what is more important is just to mention that the SODIS, you can see here, is the solar disinfection, is the use of pure sunlight without the application of any catalyst. Yes, there might be certain inactivation, but with a very long period of treatment. So the use of catalysts, the use of solar photocatalysis, using such applications seem to be quite promising. But what is more important is to see that, to find out that there might be a removal of even enteric viruses in such pilot scale experiments. So, for example, you can see that there is a considerable, I might say, removal of adenoviruses and enteroviruses. Not complete removal. No, I'm going to be honest. Complete removal is even impossible upon application of chlorination or ozonation or even UVC radiation. No, viruses are always present, are usually present in high concentrations, even after treatment, even post-treatment, especially in wastewater. That's why they should always be monitored, analyzed when we try to reuse wastewater for certain applications. And even another result here is what's going on with the resistance genes. I told you before that resistance genes maybe circulate in the environment. They are considered nowadays emerging pollutants. They can circulate in microbial populations and make bacteria resistant. That's the way they act. However, But of course, there's a broad variety of resistance genes. We cannot study every single bacteria antibiotic resistance gene. However, even in this approach, you can see that there is a considerable reduction. There might be an elimination. This is, for example, what we try to analyze. We try to analyze the free DNA, the eDNA that you can see here, and the presence of antibiotic resistance genes in phage. So you can see here that there was a presence, of course, of resistance genes. This is the presence in the effluents, but also the absence of certain genes upon usage of certain catalysts, which is quite promising. And just to give you an idea, these are the microorganisms used in solar photocatalytic disinfection studies. You can see a broad variety. It is even, you can even find literature regarding even other microorganisms. So E. coli is not the only one, no. There is also, and of course Enterococci, there is also Clostridium perfringens, a quite difficult name. However, it is a fecal indicator, anaerobic bacterium, extremely highly resistant. And it has been studied, it has been inactivated upon photocatalysis. Bacillus, other resistant genus. Staphylococcus, Pseudomonas klebsiella, as I showed you, Protozoa, and many other cases. So there's a broad variety of microorganisms. There are perspectives, considerations, limitations. Solar photocatalysis, just to conclude this presentation, seems to be quite an effective method. There is considerable inactivation of different groups of microorganisms, but special attention should be paid on the operating conditions, on the matrix that we need to disinfect. Yes, it might be successful in water and sewage samples, but upon very meticulated, let's say, setting up. So solar processes, yes, they are actually a head start in the quest of efficient disinfection treatment technologies. The whole process could be scaled up. This is, of course, a challenging task because, as I mentioned, it's quite different to work in the lab and quite different to work in the field. But of course, in the material world and research, the synthesis of new solar active and stable photocatalysts could absolutely boost the technology. But of course, special attention should be paid on the type of microorganism, the final destination, of course, of the duty stream, if we talk about wastewater disinfection. Of course, legislations, public awareness, public perceptions, but generally, solar photocatalysis may benefit from the synergy with other processes to maximize performance. So, this is the end of today's presentation. Of course, the research was performed with my collaborators, with all those institutions that you can see, the University of Patras, Hellenic Mediterranean University, the Institute of Electronic Structure and Laser in Iraklio. So I would like to thank them all. And I would like to thank you also for your attention. I hope it wasn't too tiring. Yes, it was a little biological point of view of the whole process of disinfection. Thank you all.
Thank you very much, Danai, for the presentation. So we have I know that we have overcome a little bit the time of the slot that we have for these Athena talks. However, we recognize the enthusiasm of our speaker and the very nice work that they are
Yeah, I'm
doing.
sorry, I delayed a little bit. Yes,
No, no,
I'm
no.
sorry.
There is a question. I will start with a question from the chat. One comes from our colleague George Kakavelakis, who is an assistant professor in the Hellenic Mediterranean University. Since you are working with solar light, why you don't try to match the band gap of catalysts with the maximum solar irradiation energy, for example, by desensitization of TO2? Thanks.
Yes, this is a very good question. This is a very good choice. Actually, this was my option. This was the potential to do that. I would definitely try this. But of course, I need the collaboration of my colleagues from the other departments who help me design. Of course, they design and they help me working with all those catalysts. But yes, this is a very good question and choice. I would definitely try it.
George Kakavelakis is in Hanya. He just arrived and joined us from EPFL. He's a specialist in
Oh, great.
solar cells. So I think that you can plan a meeting to come and visit you in your laboratory.
Absolutely. Oh, this is perfect. This is great. This is great because in Hanya, yes, I'm always complaining that we are very far away from anyone. So this is quite promising. Thank you very much. Yes,
Yeah,
absolutely.
I mean, I think this is a great potential.
Great.
So I cannot see any other question. I have a very fast question. Of course, I suggest you another thing. I would like to bring you in contact with Ben Gurion University in Negev in Israel. They are
Yeah.
using fantastic solar cell collectors. They have a very nice technique. But of
Okay.
course, they are using this in order to study the degradation of the solar cells. So I strongly suggest you, I will bring you in contact with the director over there.
Yes,
Go
please.
over there and
Yes.
visit their facilities regarding the solar collector, because this will help you on how to engineer much better in order to collect, the solar light for the application of your technique for in situ. However,
Yeah.
regarding, the dynamics, of course, now I'm talking and I'm going back to laboratory scale. I think that if you are going to use laser sources, like, for example, semiconductor lasers, I think that you are going to improve a lot
Yeah.
the dynamics.
We
It's not
have.
only the material, it's like
Yeah.
the source that you're using, the lasers, they provide this high dose, kind of energy delivery. So in laboratory scale, at least, you are going
Okay.
to see a much different results regarding the dynamics. I'm pretty sure.
Yes, of course, I couldn't show you everything. I have some examples of laser and certain reactors here in the labs. Yes, they are very promising. They are better results. We try to expand this knowledge here, and but just selected to show you certain nanomaterials that were synthesized from our colleagues. But yes, absolutely, we do have some results from laser, which are far more promising than those that I showed you specifically. And I'm saying promising why? Regarding bacteria, the results were very great, were perfect. But I try to focus on parasites and viruses. This is a whole different process. And not too many researchers deal with them, study them. It is not that easy. Actually, not only it is not easy, it's quite expensive. Anyway, so I try to combine, yes, the laser with the viruses and parasites. Possibly next time I could show you some results regarding them.
However, I mean, we can submit a joint, proposal. We can develop a very
Yes,
tunable laser device, let's say external cavity diode lasers.
yes,
You can tune, and the bandwidth is like less than 1 kilohertz, which means that we can tailor exactly, and we can combine what George Kakavelakis has mentioned, the
okay,
desensitized, to be closer to the energy band gap of the material that you are using, and at the same time by tuning and placing CW lasers with this line width that we're using in spectroscopy, we can activate exactly, and the efficiency will be much higher regarding the reaction.
Yeah.
And another thing that I would like to mention is like, I mean, you answered one of my questions. You're using nanoparticles, so the TO2, the metal doped TO2, they have the size of nanoparticles. Even better for lasers because due to quantum mechanics, I mean, you can tune the wavelength not by the material's energy gap only, but the size of nanoparticle finally tune, the absorptions like quantum wells. So this is something that the size of the nanoparticles also can play an important role. And I would like to ask you, in solar cells, we have observed that if we are using 2D materials as a template to place nanoparticles, the nanoparticles, they have a preferred, kind of concentration in this platform of 2D material. In this case, probably you should think if you are going to use a template like, for example, graphene oxide in order to be able to upload the nanoparticles. So the nanoparticles, the metal doped TO2, they can work as your catalysis. They are going to concentrate to these 2D platforms that combine also another effect. The size of their structures work like a filter platform. Since you mentioned that the viruses or whatever
this is what I was going to say about the virus.
I think that you can combine a double effect. I mean, probably, graphene oxide is not the most appropriate one because you have the oxygen and probably the solution processed in the water. But if, let's say, we can use graphene as a platform and over there, with some doping, some kind of doping in order, the nanoparticles to stay there, you can use, the porosity of the material to stop the bacteria, and at the same time with the light to kill,
yes. We could have double effect with graphene oxide. Exactly. Yes. This is the thing.
We will check, the bibliography with.
But not the simple, the pure graphene, as you mentioned, with the dopant, something.
Somehow you need to.
Somehow, yes.
I mean, even in solar cells, I mean, we notice that we don't have a chemical bonding, anything, but, the nanoparticles, they prefer to stay and concentrate in these flakes. Anyhow, this is like,
but this double effect is mostly wanted regarding the viral inactivation.
I remember something else. Also, probably, because you mentioned something about bacteria and the other kind of materials, I'm not a biologist. But in my master's degree, I studied and I experimented with photodynamic therapy. So we are using lasers in order to treat and create this ROS, the the reactive oxygen species, in order to kill the cancerous cells. The cancerous cells, they demonstrate some weak membrane around them, which allows, the ALA, the drug that we place, to go inside and photoactivate it and kill, the cell. So probably, some of these ideas about photodynamic therapy will help you to include more innovation in some challenges that you have in this area. Try to check, the mechanism.
Possibly, yes. It could be an option, but bacteria possess also cellular wall beyond.
But exactly this, the cellular wall, it was like a little bit weaker. Okay, the membrane of the cell.
It is quite different. That's why I mentioned. But of course we could try it. Absolutely. The cellular wall is more stable, let's say, specifically in the gram-positive bacteria. But absolutely, this is an option. We could try it. Yes. Absolutely.
Okay. So thank you very much, Danai. I think that I need to close for me also.
Thank you. Thank you very much. It was my pleasure to see you all. To see you, Kostas and Nikos. I don't know if he's here.
He just jumped out.
Okay, he just left. Okay. Thank you all.
So thank you, and we will be in contact. Thank you. Thank you, Danai. Bye.
Bye-bye.
Bye to all. Bye.